Hi, welcome everybody. Nice for you to join us. We're very excited to do this webinar with you today. So I'll explain a little bit of how it goes. So we have a pre-recorded presentation that are chopped in a few segments. So we'll show a part and then afterwards there's time for questions. You can drop your questions in the chat box. Try to do 1 or 2 questions after every segment. And at the very end, we'll have a longer Q&A. Also, I have people here, a team. Lucas is already chatting, and I will also answer questions that you have. There's also a poll that you can, there will be several polls throughout that you can answer. It will pop up a little red dot, I believe, when they're ready. And I think I've prepared everything then, said that everything needed to say. So we'll be ready to watch the first segment. Enjoy. Hello, welcome at the first of a series of webinars that we're organizing at Hudson River Biotechnology. Who am I? I'm Lotte Westerhof. I'm the R&D Innovations Director. And with me I have Lucas, Inge and Thomas, and we'll be taking you to the full CRISPR workflow. Okay, so a short introduction on Hudson River Biotechnology, so you know who we are. We're an agricultural biotech company based in the Netherlands in Wageningen. We're now over 50 employees. And first segment, if you have any questions, please ask them in the box. I'm very curious to know if these are all things that was already widely known, or if there are still a lot of questions surrounding this, or is it, what kind of audience I have. So just waiting for those. And if we have no questions about this, and the poll was already very clear that a lot of you have are struggling with regeneration, and that's definitely something that's still coming up. But one of the key points, of course, is that you really have to start with a lot of information already, and that's sometimes tricky. So do you know your gene? Can you get all the sequence information you want? Is this a fast or slow process for you? Or There's a question if the video will be available later. It will, so that's no problem if you want to review it. That's absolutely possible. Oh, there's one question I saw. Oh, actually, it is about if you need to mutate or regulate the gene, because it can still be considered a simple trait, and it is indeed still a simple trait. So usually complex traits is used for those traits that are also influenced by the environment a lot. So not only the genetics. So sometimes the genetics are very clear. Doesn't matter how good the plants are doing, they will always produce purple flowers, for example. But yeah. Then there's another question. Is it possible to manage viral disease in plant through CRISPR technology? That should be, so there are probably different strategies for that, depend on the plant and the virus, but it can be transient or stable. So there might be a natural resistance that you can CRISPR help you to doing that, but can also imagine that there's transient ways of having that, actually using the CRISPR itself to attack the virus, but it really depends on the virus. But yeah, it should be. I think we will have time for one more question if it's there. Otherwise, we'll continue with the next section. Oh, we just on the minute we get another question. You don't need any base pairs with the non-homologous and joining DNA donor inserts. So I think what you're asking, like if you have a donor DNA and you have no complementary ends, will it still get inserted? And then the answer is yes. You do not need any complementary base pairs for it to get inserted. So I hope I answered your question correctly there. Otherwise, you can always repost it and I will get back to it. But yeah, Lucas will also dive into that. It's actually quite, very high frequency. You can let this happen. All right. Then we'll continue with the next segment.
When you're thinking about which CRISPR enzyme to use, the most obvious ones are the Cas9 and the Cas12. The Cas9 has a POM sequence with a GG, and the Cas12 has a TTTV. And simply because plants tend to be more AT-rich, Cas12 is often preferred because it simply can dock at more locations in the genome as compared to Cas9. But their technical readiness is very high, so you can buy these enzymes. They know how to use, they know how the guide backbone needs to look like. There's all kinds of sites, etc. You can design your guide. They're about the same size. But, there's always a but, especially when it goes nice, the licensing costs of these enzymes are very high. And what you really have to keep in mind that there's even a bleed-through licensing cost. So if you do discoveries, like this gene does that because I CRISPR'd it out and now I'm going to breed for it naturally and not using Cas9, so I don't have to deal with the license costs. This is not true because you made the discovery using either Cas9 or Cas12, so there'll be a bleed-through cost of that discovery into your product. There are a lot of interesting variants also of Cas9 and 12 being brought to the market that are better in terms of that they have a larger genomic reach. So the POM is less needy, it just has one base pair, or perhaps maybe in the future there will be ones that don't need any POM, so you can target any place in the genome. They might be much smaller, but of course, the technical readiness for these enzymes is also much lower. So I have to wait for more experience on those if to know how to use it well. So our enzyme of choice is MAT7. So this is even outside of the Cas9 and Cas12a, and it has considerable lower licensing costs. There are more alternative enzymes out there, but their technical readiness will always be lower. For example, we also have to produce our MAT7 in-house. You cannot buy it anywhere, and we have customized software to predict guides. One more thing that is really important to understand when you are either choosing Cas9 or a Cas12 or MAT7 is that Cas9 is a blunt end cutter, so it makes a double-strand clean break, and it cuts 3 base pairs upstream of the POM, so very close to the POM, meaning you get quite small deletions, but when these deletions occur, the enzyme cannot bind again and cut again. That is different for Cas12a and MAT7, so they introduced staggered cuts, and they do that distal from the POM. So you get a tiny overhang, which is beneficial for HDR. I won't go into that too much detail now, but what is also very crucial is that it binds, it cuts, and because even when small deletions are introduced, it's not close to the POM, so it can actually recognize it again, bind again and cut again, etc. And that's for this reason, the deletions of Cas12 or MAT7 are usually much larger. And it can be a pro or a con, but at least it's much simpler for screening activity. So it's easier to know if you have a 10 base pair deletion, it's truly a deletion, and a 1 base pair deletion might be like, oh, did my sequencing shift one, or etc. So it's also differences seen with other techniques. I will go a bit into that a bit later. So, like I said, there's many tools available online to design your guides. They give you mainly 2 things: how many off-targets you can expect if it's a unique guide, and even if it's unique, what the closest off-targets would be. And some of them even give you a kind of an efficiency score. based on all kinds of research, but of course it's a prediction for both of them. Of course, the off-target is more black and white. If you really have the sequence is either there or it isn't, given you have the sequence of your variety, not the online available one. But the efficiency score is, of course, a prediction. It doesn't necessarily have to be true. The limit of these tools is they're not always present for your enzyme of choice. So for Cas9 and Cas12, they will be there. But it's not always going to be possible to select your genome of interest. And then some of these programs, if you go like, okay, my genome is not there, but a close relative is this, it'll be fine. You have to sometimes insert your sequence, and then they say this sequence doesn't occur in this genome, so then they don't run. So sometimes it's more tricky than you think. Of course, there are some workarounds, but for some you will really need to have your own program. And consider that if you select a genome in these databases, this is your species. It is not the variety you're working with. So all these SNPs that you might have in your variety is not considered in this program. So it is much better to have a program where I can upload your genomic sequence to actually predict the guides. That being said, sometimes if you just want to make a product for research sake, does this gene do what I expect it to do, and I'm not so worried about off targets, you can use these programs, of course. But the more information you have, the better. So guide design, important consideration. So what is the chance if I have a guide that it actually will be efficient? And we have a lot of experience with testing these guides in vitro, so just expose the guide or the R&P, so the CRISPR and the guide together, if they cut the target DNA, and then we always see very limited, well, we see some variation, but most of the time they work. In vivo, that is very much dependent on the transformation method you use and also the plant species. So usually we test 10, 9 out of 10 for sure will work in vitro, but in vivo can be 2 out of 10, can be 5 out of 10, can be 9 out of 10. So that's targets and species specific. And what's the chance that I do really have any off-targeting? You will have to research that. Some publications say that there's not a lot of off-targeting. Of course, it really depends on your guide. But some publications state that the normal mutation rate is actually higher than the off-target events. But again, there needs to be more research into this, because it also depends on the transformation method that you use. I will go more into transformation methods, but we do it transiently with very high concentrations, and there's a lot of methods that do it stably with very low concentrations. And at this moment, we don't know which actually gives more off-targets. So this is an example of one of those in vitro screenings of the guides. So there's a linear piece of DNA with the target, and then we tested here, I think it was 30 guides that cut in different places, resulting in 2 smaller bands. And then you can see for most, this top band is gone, for some not, but these 2 bands appear almost everywhere. So the variability between efficiency of these guides is, you can kind of see on how much of this top band is still there.
Hello, everybody. I already have 2 questions, so that's super nice. Somebody was wondering about the in vitro testing, what I actually mean by that. And of course, these in vitro, in vivo, it's sometimes used interchangeably. But so when we really make the CRISPR enzyme and we buy the guide, actually, you can order that, and we combine them in the lab. Make sure they form a complex, and then we expose them to the linearized target DNA. So this is really in a test tube, and then we run it on gel to see if their cutting has occurred. So in vivo, I really mean in the plant. All guides that they're predicted, they should cut. And then there was one more question about if a gene can be deleted also without adding another gene. So in this sense, it's like, yeah, so you can delete a whole gene or a simple knockout, but you can also delete a whole gene. And then if you do it indeed in the way we do it without transgenes, then you're not adding any other gene. You could also do it with transgenes and then cross them out, which and then the resulting product also doesn't have an extra gene. But yeah, and to delete a whole gene, you will need a minimum of 2 guides, but usually a couple of guides to make sure the whole fragment is gone. Let me see. There was another one coming in, I think. Indeed, about whether or not you can call certain things a GMO or not a GMO. So indeed, if you don't use any extra DNA, no donor DNA, only the CRISPR enzyme, and it makes a break for you and it repairs, and there may be a few base pairs deleted or even inserted, or even if it's a bigger deletion, in a lot of areas, this is not considered a GMO. But in Europe, it still is. We hope and expect this to change in the future, but as it is now, it is considered GMO in Europe, although it is from the lowest tier. So there's different tiers for how severe this GMO is. We would say whereby the tier 3 is really adding a gene from another realm into it, and tier 2 is adding DNA that's from a related species into it. I hope that answers your question. And then indeed, even if you do only a deletion with the CRISPR, it is a case, depending on the country you're in and what the deletion exactly looks like. But most of the cases, it would not be ruled as a GMO. I think I answered all the questions. If not, or I didn't answer them fully, please respond again, and I will dive back into it. The next segment is about finally how do we know what we want to edit, we know what we want to do edit, we know what CRISPR enzyme we want to use. So now how do we get this all inside the plants to do its job? And I will dive into the 2 main flavors of the different strategies.
So how do you get your CRISPR cargo into the cells? I share with you a picture of a very traditional transformation method. So we use this a lot on model plant species with agrobacteriums, transferring transgenes into the plants, and these transgenes can code for the CRISPR enzyme and your guide. So then the transgenes are introduced into the plant cell makes these products, the RNP assembles inside the plant, so the CRISPR enzyme in the guide, and then finds its target, makes the double strand break, and then the plant repairs. When you're going through this process, you regenerate the plants and the transgenes will be there. The nice thing about that, you can also select for it, but the not so nice thing is that you will always get heterozygous product. So because undifferentiated cells form a new plant, but they might not arise from one cell. So they might be a mix, one having the transgene, the other not. And then even if they have the transgenes, some might be edited, some might be not, some might be edited over time while the plant grows. So you will always have to clean this up. And this is why we chose for this approach, so the RNP-based approach. So you make the RNP outside of the plant cell, and then you take the plant and we make protoplasts out of that. So we degrade the cell wall and we have the individual cells, the protoplasts, and we transfect that with the RNPs. You can do this in different ways. PEG is a very standard way. So you make little holes in the membrane, allowing the RNP to go in and find its way to the nucleus and do the editing there. and then we combine that with single cell regeneration. The nice thing is that the RNP gets degraded inside the cell at some stage, so there will be no transgenes, no more CRISPR enzyme, just the one cell that either got edited or not, but we have single cell regeneration of those, and you can test in a later stage whether or not the plant was edited. But if it was edited, it's a clean homozygous plant. So I told you about these two flavors and going a bit more into what their advantages and disadvantages are. So you have transgene mediated, can be via agrobacterium, but can also be bombardment. Agrobacterium can also be applied in different ways, either via explant or floral dip. All these methods are transgene mediated. Their advantages is that it's very technologically ready. You can use selection markers so you know where your transgenes are. The disadvantages, however, that it requires cloning for every guide. So for every target, you need to have a transgene. Regeneration is a challenge, so you need to be able to have this procedure in place to be able to do this, and it's a very long time to market because indeed it produces these heterozygous plants that you need to clean up, then you need to cross out the transgenes and bulk up the product. Also, there's the burden of proof that this plant, the product that you have in the end, is no longer a GMO. So the transgenes are gone, 100%, and only the edit remains. So the RNP-based method, you do the protoplast transfection, either chemical or electroporation, or nanoparticles can also be used. And the advantage is that there's no cloning required, so you either produce or buy your CRISPR enzyme, you can buy your guide, and you mix these, which is very controllable. So even if you want to multiplex several guides, and then you mix these all first independently, and then when you do the transfection, you combine them. It's super scalable, especially when you combine it with single cell regeneration. And the great thing we think is that it's DNA free. So I mean, there's no burden of proof. There's also a downside to this technology. So it's very technologically complex. Of course, we've spent already a couple of years trying to really perfect this way. It's not easily done. If you don't know how to make protoplast of your species, it's tricky. The editing screening, if you could do that very fast, you can go through a lot of rounds, but if that's a long process, it's very complicated. That being said, also the regeneration for protoplast is definitely also a challenge. So even maybe more so than the explant regeneration, because you have to go also from protoplast back to callus and not only from callus to shoots back into a plant. The nice thing about that though is that also this is very scalable. If you have a regeneration challenge and you have all these different, protoplasts, are a lot of protoplasts, you can sort them, you can test all the different conditions they need. And also if you have a lot of different calli, clean, edited, homozygous calli, you can do a large scale throughput screening of which conditions are conducive to the regeneration. Okay, let me take you a bit more on board with the controllability of the Tiger workflow. So the Tiger workflow, we do transfection of protoplasts, and this pool of protoplasts we can assess with flow cytometry of amount of viability. So how many of these protoplasts are viable and how many of them receive the MAT7 protein, as we have this coupled to a fluorescent protein. So we have the viability and we have the amount of MAT7 going in, and then we can couple that later with the editing efficiency. These are microscopy pictures also of the MAT7 with the GFP attached over time, and then you can really nicely see the shuttling of the MAT7 GFP into the nucleus of the cell, where it obviously needs to do its job.
Hi, welcome back. So I got a questions about patent rules concerning Cas9 use. Unfortunately, I cannot give a very satisfying answer to that probably because it's very complex. It's evaluated case by case. Do not believe it's easy to pass on the freedom to use company to company. We use MUT7 for this purpose, so we generally don't need to worry about that. So cannot give you a more clarifying answer there, I'm afraid. We got another question about when delivering RNPs, which method is preferred in your practice, PEG or electroporation? This depends on the plant species. So far, we've gotten PEG always to work, but sometimes electroporation can be more efficient in some species. But I think both will result in a positive transfection. Let's see. There are some polls also out. Please, if you can fill them in, would be highly appreciated. So there are so many different ways of transfections, and all of them are perfectly fine and really depend on the plant you're working with, which is probably the most suitable, and all the capabilities and how much effort and expertise you want to put into the endeavor. So there's a lot of pros and cons. So this is the main flavors, right? Transgene free or with transgenes. If you can easily cross them out, transgenes are very attractive. The next bit is about screening for editing. And there it's really relevant that we keep in mind of when we say editing efficiency, what we're talking about. But I will dive into that a little bit. Just get another question pop in, see if I can give that a quick read to see if I can answer that still. This is a very particular one. It's about resistance to viruses, and indeed there are ways to breed and to CRISPR for virus resistance. But I think I'm gonna, while the next segment runs, read that a bit more thoroughly and hopefully answer that. Coming up.
What remains is still doesn't do its job, so the screening for editing. So there's a lot of methods for screening, especially from the pharmaceutical world. There's a lot of different ways to do it. In the plant world, what is mostly done is either PCRs or sequencing, and then you have a lot of in-between still. The advantages, of course, of the PCR is that it's simple and it's cheap and it's fast. Of course, the disadvantages is that you don't have all the information. So when you sequence, you know exactly what the edit is. With PCR, you can either see a drop in the size of the PCR band, meaning that you got a very, very large deletion, but of course it needs to be more than even 10 base pairs. So you need to either use 2 guides to ensure that you really get a significant drop, or you target a place in the genome that would naturally have a restriction site, and then you do the PCR and then the restriction, and if the restriction site's no longer there, you may assume that it's been edited. But also that is, of course, if you don't see something, it's not so nice as if you do see something. You have alternatives like high-resolution melt curve. So that means that you do the PCR with a qPCR machine and then see how fast the fragment falls apart and if it's in a different melting temperature than your original amplicon. It's quite a nice technique. Of course, it needs you to have a qPCR machine, and you need quite high amounts of DNA to edit this. And then you have a lot of more intermediates that I'm not going to go into because they will all have the same disadvantage, that you either need specialized equipment and that they don't give you the full information that sequencing does. So we will always go for amplicon sequencing. The nice thing is that it gives you indeed complete information on how the deletion or an indel looks like. And it is very fast if you have the technology in-house. If you don't, then you have to send these amplicons out to sequencing, and that turnaround time can be quite long. But hoping that get better in recent or in the coming time. The nicest, obviously, is full genome sequencing, so you have all your information about your targets all at the same time, or all of your off-targets as well. Of course, it's expensive and it requires some extensive bioinformatics, but it's really nice, especially for your final plant or your product that you want to do. It's almost a prerequisite there. And you need a lot of ultra-pure DNA for that. But yeah, when you get to that stage, you're probably very happy to do so. To give you a bit of a snapshot of how this looks like for us when we're editing with MAT7 in a plant. So this is an example where there's 3 different guides indicated, but they were either not used or 2x2. So you have either the 272 combined with the 334. I should have renamed these anyway. So you see, this is a collapsed read whereby the dark gray indicates missing nucleotides and the light gray nucleotides that are there. And then you see that at both guides location, about 60% of the cases, the base pair surrounding that site have disappeared. And also in between, 35% of the time, the whole fragments between these guides, targets were lost. And the same is for this example in the middle. So it's 45 and 49% editing of the individual guides and 33% of the fragments. So these are, when we transfect the protoplasts and we do an amplicon sequencing, these percentages reflect the number of reads that either have or do not have these base pairs. This is not, us having individual clones and then 57 out of the 100 clones not having this fragment. So this is on the mix. So this is what we do as a pre-screen to know whether or not we want to continue with a specific guide or an editing strategy. But that's very good to remember sometimes when we're talking about editing efficiency, that we're not comparing apples with pears all the time. So is it either a mix or your plant, or if you have a clone, do we talk about 40% editing in all the cells of the plant? Or are we talking about 40% of the plant? Or are we talking about 40%, like 4 of the 10 alleles? Or, right? So really have to always describe what we're talking about. So yeah, that's actually what so transient versus the clone, and then the number of clones or part of the clones, or the number of copies and alleles.
Hi, just checking in if there's any questions about this. I know there's many more methods of detection, and they all probably have their advantages and disadvantages. Amplicon sequencing simply gives you the most information. Often you can send out your amplicons for sequencing. The only downside that it's really, has a long turnaround sometimes for you to get back the data. Surely that will get better over time. I don't expect that to take very long for that to get better. And when you can do it in-house, it's perfect, of course. It's nanopore sequencing is relatively easy to set up in-house. All is relative, of course, but it's something to consider if you really want to do this. I didn't see any questions pop up. This is also a quite short fragment. There's a little wrap-up segment still. I realized that there's not too much about regeneration in there. I can follow up a little bit with that at the end. And also when there's some longer Q&A question. If you have any questions about that already, you can already put them in. But then we'll go ahead now with sharing the last segment, and then we can wrap up with a longer Q&A.
All right, and then I want to go back to reaching homozygosity. So again, this comparison between transgene methods and the Tiger workflow. So transgene methods, you have to select for the transgene, but you might end up with a plant that's not completely homozygous because it's always emerging from several cells, and the Tiger workflow where it all arises from one cell. So if it's from one cell, it's very simple. You just have to confirm that it was really from one cell, and with the regeneration with transient, you really have to grow the plant, do a couple of cuttings, or even go to seeds and then evaluate the offspring to be 100% sure you have a genetically stable product. So I want to go a bit more into the scalability of the Tiger workflow. So you have from a single cell immediately homozygous plant. But how do you know it's really from one cell? We always need to check that, but we embed our protoplast in such a low concentration that the microcolony that arises from that, the chance that it's being a single cell is very, very big. And then we plate that out in a 96 well plate. We're even automating this now and using microfluidics to put one cell per well even. It's very hard to grow these cells in such low concentrations, but we found ways to do that. So just want to show you our Tiger workflow, how it would work manually. So I already mentioned we're automating all these different steps, but you have at the first moment, at 0 hours, you have your protoplasts. They start dividing or remaking their cell wall and start dividing after 2 days. 2 weeks later, you can already distinguish a microcolony. And after 4 weeks, you can see them with the naked eye. And then they're cut out of their discs and plated. And they're cultivated for a certain amount of time. And then the shoot induction starts. And then they really started to be moved to bigger plates and treated as individuals while we do the screening and the shoot isolation. So sometimes we do the screening already on callus, sometimes we do the screening only on the shoot. That depends a bit on the editing efficiency we expect and the shooting efficiency that we're having for a particular species. But always we also screen the protoplast pool that goes into the experiment, so we can predict actually the editing efficiency in it. So all of this being automated, but this is how you would do it manually. Of course, what we should not forget that once we have our plant, we really need to do a phenotypic screening. Did we get the trait we were going for? Or did we get also things that we didn't ask for, like our CRISPR off targets? So here's where you would want to really sequence the whole genome. And what you always want to take along during your procedure is also untreated cells. Mock transfected or not transfected at all, to see if you would have any changes from the wild type just by going through all the tissue culturing. So really want to, there can be tissue culture effects on epigenetics, but also some chromosomal rearrangements or influence on ploidity can occur. So you have to, for sure, make sure that you also aim for not getting maybe one mutant, but several, and really comparing that to your untransformed or your mock transfected controls when you're really looking into the phenotype. And with that, I want to conclude this webinar. I'm very open for questions, and I hope you enjoyed it and it was all clear. If not, please ask your questions.
Hi, hope you enjoyed that indeed. So we have a bit longer now for the Q&A, and a question pops up if we use alginate to culture our protoplast. And we do. This is indeed one of the most classic materials, but this is also immediately what I wanted to say, that I can elaborate a little bit more on regeneration, is that we also have a lot of other embedding materials than alginate that seems to do much better. So while alginate's perfectly fine for some species, it may not be for others. especially when you have phenolic compound production. They really seem to not be able to leach out very well from the alginate, and they might end up killing your precious little clone that you have in there. So that's one of the things that we work on, and that we also materials that can be automated easily. We have projects that we set up all the protocols for a given species and the varieties of that species. And then we usually also do, we start off, we set up all the protoplasting protocols, but we immediately also do a lot of explant trials to see, okay, how can we get this material to make colors the most easily? How can we get those colors to shoot easily? Or can we induce embryonic colors? And also determine a little bit the biological potential, how easy is this going, especially if you have within the same species a lot of varieties. Not all varieties will respond equally, or also part of the research is like, okay, if we develop a protocol for a certain variety, how easily could it be adopted to another variety? There's always a lot of overlap between varieties of a certain species, but they're definitely not all, plug and play. Once you know it for a certain species, that you can always do it. I get a question about cost. So of course, so we have a lot of projects doing this for other companies. If you are interested, you can contact us via the website or via LinkedIn. You can find us, and people will be in contact with you and happy to discuss all the details and immediately get all the questions that's really relevant for you, and it can give you quite quickly an idea of what we can do for you. We are completely independent, so we can work for anybody. We don't have any shareholders. We do not have any wish to be a seed or breeding company. So we're really a technology provider, not a service provider. So we do really have long-standing relationship with clients and do research for them, and we use all the technology and capability that we have at our disposal to set this up for our clients. And usually when one project is done, there's a new project, etc. That's not a prerequisite, of course, but just we're a bit different than some companies that we have. We cannot work in certain crops or anything, so we can work for anybody in any crop. Yeah, there's the questions. Any technical preference in working with Cas9 or working with CPF1 nucleases in general in plants or in specific crops? So the really nice thing about the CPF1 or the 12As is that the palms are more suited to plants because it's more AT rich, and it gives bigger deletions, which is quite nice for screening. And then we use MAT7, which is also like that, and it's of course also much better in terms of licensing cost. So we prefer MAT7, but on other fronts, there's no real difference. So if you can have the Cas9 pom-pom is NGG, so if you can find a GG at the location that you're interested in, why not use Cas9 if you're willing to pay the license fee anyway? There would not be a real preference. So I'm just seeing if more questions pop in, and I can think about things that I might not have included. So the regeneration, I know a lot of people are struggling with that, and it is an ongoing struggle also for us. There's always some species that you cannot do yet, but that counts for all methods. So if there is not a regeneration protocol, it's really hard to do this. And yeah, we tackle this in different ways to use advanced materials for the embedding, and doing this really high throughput with all the latest developments on regeneration boosters, also from the pharma world. And there is trickling in knowledge obviously about genes that really influence, and how can you get those genes switched on at the right time, or their gene products delivered at the right time to facilitate the shooting or the callus induction. But yeah, once a protocol is there and it works, then it's again being able to do this very high throughput and on a scalable manner. And that's what we do here. Type of traits. So we get a lot of questions about what plants we work in and what type of traits. We cannot divulge all of this, but there's we work with more than 15 crops. I think we added another one just now, and there's some ornamentals in there, but also some vegetable and some row crops, and there are all kinds of types of traits. Obviously, some ornamentals you can usually disease resistance or something to do with how beautiful they are. And yeah, we also have, I have to see what I can say that's vague enough. Yeah, so also increase in yield or stuff like that. Yeah. What type of protoplast do we prefer to work with? So yes, and there is indeed always the risk of somoclonal variation. So we like to work with leaf protoplast as a first start. We think is the shorter we can have the protoplast and the regenerating calli in tissue culture, the better. If the leaf protoplasts do not work out, then indeed we would go maybe via callus to first have them dedifferentiate and then use, or embryonic callus. But that can take some time to get established culture. And of course, it's always time that we are shy of, and we always want it faster. So our first trial is always leaf. It's easiest to get by. I hope I gave enough hints on the regeneration. We have a lot of regeneration specialists in how that can tell you much more about it than I can. So even if you have questions there or interested anyway, don't hesitate to contact us and we see what we can do for you. The video will be available after this, so you can have another look at everything if it went too fast. And I wanted to thank you very much for joining us. And I'm going to have a look at my colleague if there's anything I forgot to say.
We can check out the link that's in the chat if you want more information.
Yes, right. There's a link posted in the chat, and you can click on that if you want some more information. And oh, I just got another question. I'll obviously still answer that. Do you work on onion? Not yet, I can tell you that. And indeed, it is, of course, a nightmare for sequencing because it's quite big. But they say asking if there's a suitable primer, yeah, that's really dependent on your target. So I have difficulty answering that. But whole genome sequencing for onion is a challenge. And also the regeneration, I understood, it's quite a challenge. Huh? But up for it, always up for it, yes. Yeah. All right. I will leave it at this. I wanted to thank you so much for joining us and taking the time to check out this webinar with us. I hope you enjoyed it. I hope everything was clear. If not, contact us via the link, and have a wonderful